So now today's topic is translation. In the process of translation, that's a formation of protein from mRNA with the help of राइबोसोम। राइबोसोम की हेल्प से हम प्रोटीन सिंथेसाइज करेंगे जो mRNA कोड लिए हैं डीएनए से। इसके लिए हम इसको कुछ स्टेप्स में डिवाइड करेंगे। फर्स्ट स्टेप है, फर्स्ट इज एक्टिवेशन ऑफ अमीनो एसिड। We know very well there are of 20 different types of amino acid participate in protein formation. These amino acids now react with ATP. Amino acids with ATP to form amino acyl AMP enzyme complex. Amino acyl AMP एंजाइम कॉम्प्लेक्स। अभी एमिनो एसिड एएमपी एंजाइम कॉम्प्लेक्स आया किस वजह से, जिसे हम बोलेंगे, कह सकते हैं हम एक्टिवेटेड अमीनो एसिड। तो इसके लिए हम क्या कह सकते हैं कि जो भी हमारे पास अमीनो एसिड था, 20 अमीनो एसिड्स हैं, कोई भी एक अमीनो एसिड है हमारे पास। इसके बाद ये हो गया एटीपी, ये फॉर्म हो गया अमीनो एसाइल एएमपी एंजाइम कॉम्प्लेक्स प्लस पीपी बेटा। पीपी का मतलब क्या हो गया यहाँ पे, जो फॉस्फोरस थे दो, जो एटीपी है, इट ब्रेक इनटू एडीपी एडेनोसिन डाइफॉस्फेट, और इट आल्सो ब्रेक इनटू एएमपी मोनोफॉस्फेट। तो एक फॉस्फेट ग्रुप यहाँ से रिलीज होगा, एक फॉस्फेट यहाँ से रिलीज होगा, ये दोनों फॉस्फेट हो गए यहाँ पे। इसके अलावा ये एएमपी जो है ये अमीनो ग्रुप से अटैच हो जाएगा। इसका मतलब अमीनो ग्रुप अभी क्या हो गया यहाँ पे, एक्टिवेटेड हो गया। नेक्स्ट इसमें आएगा, इसके लिए जो हमने एंजाइम यूज किया, वो यूज किया हमने अमाइनो एसाइल टी आर एन ए synthesis, or in some book it is somehow to written as amino acyl synthetase enzyme। हर एक अमीनो एसिड के लिए अलग स्पेसिफिक अमीनो एसाइल टीआरएनए सिंथेसिस एंजाइम प्रेजेंट होगा. We just mention it here. Reaction catalyzed by specific amino acyl tRNA synthetase enzyme, or there is a separate amino acyl tRNA synthetase for each kind of amino acids. A particular amino acid was activated by a specific tRNA amino acid synthetase enzyme here. Now, second step is charging of tRNA. Guys, tRNA is an important role for the formation of protein, or we can say activation of tRNA. Charging of tRNA or activation of tRNA, or we can also say loading of tRNA. Loading of tRNA. The very important thing is that specific amino acid recognized or analyzed or identified by specific tRNA means a particular means we have 20 type of amino acids, means we have 20 different type of tRNAs here. Okay, now what happened, अब जो हमने एक्टिवेटेड अमीनो एसिड यूज किया था, तो हम यहाँ लिखेंगे अमीनो एसिड अटैच टू अमीनो एसिड अटैचमेंट और बाइंडिंग साइट. अमीनो एसिड अटैचमेंट साइट और अमीनो एसिड बाइंडिंग साइट ऑफ tRNA and AMP adenosine monophosphate and enzyme are separate out. अब यहाँ पे क्या हुआ, हमने ऊपर देखा था अमीनो एसिड, वेट बच्चा, अब हुआ क्या, हमने ऊपर अभी लास्ट स्टेप में बात करी थी अमाइनो एसाइल एएमपी एंजाइम कॉम्प्लेक्स प्लस टीआरएनए. तो अभी हमने क्या एनालाइज किया यहाँ पर कि जैसे ही अमीनो एसाइल एएमपी एंजाइम कॉम्प्लेक्स टीआरएनए से अटैच होगा तो यहाँ पे एएमपी और एंजाइम सेपरेट आउट हो जाएंगे। जब एएमपी और एंजाइम सेपरेट आउट हो जाएंगे तो क्या होगा, टीआरएनए अमीनो एसिड के साथ बाइंड हो जाएगा। तो हम यहाँ लिख सकते हैं अमाइनो एसाइल टीआरएनए कॉम्प्लेक्स प्लस एएमपी, this one, plus enzyme, these are separate out. Now amino acid attached with tRNA, or amino acid acyl tRNA complex is also called as, I mention it here, charged tRNA. Now this charged tRNA moves toward ribosome. Now the third step is formation of protein or translation, formation of protein or translation here. Now there are few things which initiate it. First step we included here as initiation of polypeptide chain. Means now polypeptide chain start to form. Initially we have few things here. Ribosome with 30s unit with 50s unit. We have GTP, guanosine triphosphate. It is equivalent to ATP here. Magnesium ion concentration means it is essential for the formation of polypeptide chain. Charged tRNA or amino acyl tRNA complex, and the most important thing is mRNA. With this we have some initiation factor. These initiation factors are different in prokaryotes and eukaryotes. We just noted here prokaryotes initiation factor. The prokaryotes initiation factor for this are IF1, IF2, IF3, and these initiation factor for prokaryotes, they are of some specific type of proteins. Specific specific proteins, and energy in the case of or in the form of GTP, energy in the form of GTP plus initiation factor proceed or promote initiation process. Now, one thing in prokaryotes also, it's an important thing or important factor, that is in prokaryotes. With the help of SD sequence. SD sequence means sign Dalgarno sequence. Sign Dalgarno sequence or SD sequence. What the meaning of this? It's a sequence. It's a sequence of 8 nitrogen bases is present before the 4 to 12 nitrogen bases of initiation codon on mRNA. Means when we are going to talk about mRNA, just for mRNA, we are just assume it's a mRNA sequence. Before mRNA sequence, there is a starting codons or initiating codons, means AUG, repeated unit of AUG. And before this AUG, there are the 8 nitrogen bases of a specific sequence are called SD sequence. This we are going to talk about mRNA. Means we analyze the mRNA. Now, how can mRNA analyze ribosomal sequence? So this sequence, the similar subunit of ribosome, this I just noted here, in similar smaller subunit of ribosome have complementary sequence of SD sequence called anti-sign Dalgarno sequence. Anti-sign Dalgarno sequence. I noted here. Anti-sign Dalgarno sequence here. With the help of SD and ASD. With the help of SD and ASD sequence, mRNA recognize the smaller subunit of ribosome. What is the meaning of smaller subunit? That is, we noted here. Ribosome made up of 30S and 50S. 30S means it's a smaller unit, and a 50S means it's a larger unit. So I mention it first of all. So we just noted here, that is SD and ASD sequence. These both sequence are get together of mRNA recognize the smaller subunit of ribosome. We are just talk about prokaryotes. Now what will happen in eukaryotes? Here smaller subunit of ribosome is recognized by 7 mg cap, means the cap formed, that is a methyl guanosine triphosphate cap, or 7 mg cap, which recognize a smaller unit of ribosome here. In eukaryotes, 18S rRNA, ribosomal Na, 18S ribosomal RNA, or sorry, uh, smaller subunit has complementary sequence of 7 mg cap, means 30S, that is smaller subunit, plus mRNA initiating factor IF3 with magnesium ion concentration 30S mRNA complex. This complex is formed here. Now this now guys, this 30S mRNA complex reacted with formyl or formyl methionine tRNA. Formyl methionine tRNA means activated tRNA complex, and these both of made up of 30S mRNA, 30S mRNA, formyl methionine tRNA, formyl methionine tRNA complex, and the whole process required energy, energy in the form of GTP and some initiating factor IF2, IF3, and magnesium ion concentration in this step. Now, large subunit, that is 50S, join this complex. The initiation are factor released and complete 70S ribosome is formed here. Complete as 70S ribosome formed. Now, There is a ribosomal unit, large unit, small unit. Here, small unit runs along with mRNA and large sites for tRNA. First, P site. P. Second is A site, and third is E site. And make it as a point. A large subunit of ribosome. There are three sites for tRNA. What are these three sites? P site means peptidal sites where charged amino acid entered. Second is A site means amino acyl site where amino acid form. Third is S site means exit site means uncharged or without amino acids tRNA. We just mentioned some points also. The starting codon of mRNA. Starting codon of mRNA is near to P site. Means if there is a mRNA starting site, there's a P site here starting. Next one is tRNA with formyl methionine amino acid. First, attaches to P site. Firstly attached to P site of ribosome here. Means tRNA with amino acid firstly attached to P site or entered toward P site. And next codon of mRNA. Next codon of mRNA is near to A site. Means firstly P entered here, then A, after that E. So next new tRNA with a new amino acid with always attached to A site. A site of ribosome. Okay, but initial means if it's a we assume it's a A1, another A is A2. Initial A site is empty. How could it possible? We firstly assume or assumption that it's a tRNA means it's a T, it's a amino acid or amino acyl 1 A1. We mark it as a A1. We just assume there's a 5, there's a 3. We mark it as a system of 3 codons. Here, small subunit, large unit, AUG. A1, anticodon of tRNA attached with this, and this side it's entered is a P site. Now A site is empty here. It's a site. And next of all, what will happen? 5 prime, 3 prime, A1, it's a A2. Means it's a P site or it's a A site. It's a exit site E site. A1, A2. Here A2 is there. ठीक है? In the next step, what will happen? This A1 just mark about to A2 site. Now the third one, chain termination. Due to sliding of ribosome along mRNA when any non-sense codon, that is UAA, any one of these three, sorry, UAG and UGA at A site The process of translation stopped. The linkage between the last tRNA and polypeptide chain broken by these three release factor, means any one of these release factors which are RF1 means releasing factor 1, RF2 releasing factor 2, and RF3 releasing factor 3, with the help of the energy with taken to GTP here. This process of the whole stopped, and the now in this sense these are called as untranslated region. We mentioned it here. And mRNA also have some additional sequences that are not translated, are referred to as untranslated regions. And these untranslated regions present on 5 dash end before starting codon. Before starting codon, or 3 dash end after stop codon. And this required means UTR untranslated region present on mRNA are required for efficient translation process, means by the recognition of ribosomal sites here.